Bilirubin metabolism starts with bilirubin formation. Bilirubin is formed by breakdown of heme, which is present in hemoglobin, myoglobin, and others. Bilirubin is generated by sequential catalytic degradation of heme in reticuloendothelial cells, which is mediated by two groups of enzymes. Heme oxygenase initiates the opening of the porphyrin ring of heme by catalyzing the oxidation of the alpha-carbon bridge. This leads to the formation of biliverdin, which is then reduced by biliverdin reductase to bilirubin. The next step is binding of bilirubin to plasma albumin. Bilirubin is very poorly soluble in water at physiologic pH. Bilirubin reversibly binds to albumin. And this albumin binding keeps bilirubin in the vascular space, thereby preventing bilirubin deposition into extrahepatic tissues, and transports bilirubin to the sinusoidal surface of the hepatocyte, where the bilirubin dissociates from albumin and enters the hepatocyte for conjugation. After reaching the liver, hepatic uptake and storage of bilirubin occurs. In the liver sinusoids, the albumin-bilirubin complex dissociates, and the bilirubin is taken up efficiently by the hepatocyte while the albumin remains in circulation. Bilirubin is taken up by hepatocytes by a process of facilitated diffusion, which is not energy-consuming. As a result, transport cannot occur against a concentration gradient, and is bidirectional. Sinusoidal bilirubin uptake thought to be mediated by carrier proteins that have not been fully characterized. Within the hepatocyte, bilirubin binds to glutathione S-transferases, GSTs. GST binding reduces the efflux of the internalized bilirubin, thereby increasing net uptake. A fraction of the unconjugated and conjugated bilirubin within the hepatocytes is transported back into the sinusoidal blood. This fraction undergoes reuptake by hepatocytes downstream to the sinusoidal blood flow. The reuptake is mediated by two proteins, organic anion transporter protein 1b1 and 1b3, OATP1b1 and OATP1b3. One of the most important steps in bilirubin metabolism is conjugation of bilirubin. Glucuronidation is mediated by a family of enzymes, termed uridine diphosphoglucuronate glucuronosyltransferase, UGT. Glucuronides are more water-soluble and are readily excreted in bile and urine. Of the various isoforms of the uridine diphosphoglucuronate glucuronosyltransferase, UGT, family of enzymes, only one isoform, UGT1A1 is physiologically important in bilirubin glucuronidation. UGT1A1 is an intrinsic protein of the endoplasmic reticulum, and its catalytic site is located within the lumen. The ultimate goal is excretion of conjugated bilirubin. Active transport into the bile. Conjugated bilirubin is actively transported across the bile canalicular membrane of the hepatocyte against a concentration gradient. This canalicular secretion of bilirubin occurs through an organic anion transporter, termed multi-drug resistance protein 2 or ATP binding cassette, ABC, C2. A portion of the conjugated bilirubin is secreted back into the sinusoidal blood through another transporter named multi-drug resistance protein 3, ABC, C3. Most of the conjugated bilirubin secreted via this transporter is reuptaken by hepatocytes downstream to the sinusoidal blood flow through the sinusoidal surface organic anion transporters called OATP1b1 and OATP1b3. After secreted into the bile and reaching the digestive tract, degradation of bilirubin occurs. Conjugated bilirubin is water-soluble and is not absorbed across the lipid membrane of the small intestinal epithelium. In comparison, the unconjugated bilirubin fraction is partially reabsorbed and undergoes enterohepatic circulation. Bilirubin is reduced by bacterial enzymes in the colon to a series of molecules, termed urobilinogen. It is partly absorbed in the bowel and undergoes hepatobiliary recirculation. The two major urobilinoids found in stool, urobilinogen and stercobilinogen, are colorless and turn orange-yellow only after oxidation to urobilins in the gut. Excretion by the kidneys. The fraction of urobilinogen that is not cleared by the liver enters the general circulation and is partly excreted in urine as urinary urobilinogen. Unconjugated bilirubin is not excreted in the urine normally, because it is bind to albumin.